Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Many people know mebendazole is a pharmaceutical medication for parasites. But over the last several years, researchers have been asking a very different question. Could this old anti-parasitic drug also have anti-cancer effects? And not just through one pathway. In preclinical studies, mebendazole appears to interact with several systems cancer cells depend on to grow, invade, build blood supply, and resist cell death. This makes mebendazole scientifically interesting as a repurposed drug candidate. So in this video, we're going to look at what mebendazole is, why researchers are studying it in cancer, and the major non-metabolic mechanisms in which it may have anti-tumor effects. So without further ado, let's get into it. This video is meant to be an overview of mebendazole and what it is and how it potentially kills cancer cells, but looking at it from a more generalized or a more classical point of view and not really looking at the deeper metabolic changes that happen with specifically glucose and glutamine metabolism, which we will look at in subsequent videos. The first paper that I'm going to pull from was published in August of 2019 and is titled Mebendazole as a Candidate for Drug Repurposing in Oncology, an extensive review of the current literature. And it says here, mebendazole meets many of the characteristics desirable for a repurposed drug, good and proven toxicity profile, pharmacokinetics allowing to reach therapeutic concentrations at the disease site, ease of administration, and low price, or it should be a low price, not always the reality. Several in vitro studies suggest that mebendazole inhibits a wide range of factors involved in tumor progression, such as tubulin polymerization, angiogenesis, pro-survival pathways, matrix metalloproteinases, and multi-drug resistant protein transporters. Mebendazole not only exerts direct cytotoxic activity, but also synergizes with ionizing radiation and different chemotherapeutic agents and stimulates anti-tumor immune response. In vivo, mebendazole treatment as a single agent or in combination with chemotherapy led to a reduction or complete arrest of tumor growth, marked decrease in metastatic spread, and improvements in survival. And I included these chemical structures at the bottom because this is all part of a family of medications that we will be talking about in this series. I'm going to start with mebendazole, and then I'm going to hit fenbendazole, and lastly, due to audience interest, I'm going to also hit albendazole as well. So I broke this paper down into its kind of sub-chapters, and it goes along nicely with this graphic that was also part of this paper, which is looking at tubulin depolymerization, angiogenesis and inhibition, inhibition of signal transduction pathways involved in cancer progression, sensitization of chemo and radiotherapy, induction of apoptosis and cytotoxicity, inhibition of kinases, and induction of anti-tumor immune response. And so each place there is a kind of red lightning bolt, there is an inhibition of that process, either by decreased expression or by direct inhibition. And then anywhere you see a green line, then mebendazole is having a induction or an activation effect at that level. And so I'm going to start with the top left here and look at angiogenesis. So here it is inhibiting VEGF or VEGF receptor interaction. There's inhibitions of matrix metalloproteinases. There is inhibitions of BCL2 and anti-apoptotic factors, increases in cytochrome C and the major caspases involved in apoptosis, as well as many of the diseased signaling pathways that ultimately end up with upregulated glucose and glutamine metabolism via metabolic reprogramming, such as MYC and other pro-growth pathways. Then, as stated in the paper, there are effects on these anti-cancer drug efflux pathways, effects on M1 polarization, as well as probably the most well-known mechanism, which is the tubulin or microtubule polymerization, or the building of microtubules and inhibiting that process as well. The next paper we're going to look at was published in January of 2023 and is titled Emerging Perspectives on the Anti-Parasitic Mebendazole as a Repurposed Drug for the Treatment of Brain Cancers. And it says here, mebendazole can penetrate the blood-brain barrier and has been shown to inhibit the malignant progression of glioma by targeting signaling pathways related to cell proliferation, apoptosis, or invasion migration, or by increasing the sensitivity of glioma cells to conventional chemotherapy or radiotherapy. Moreover, several preclinical models and ongoing clinical trials explore the efficacy of mebendazole in multiple cancers, including AML, acute myeloid leukemia, brain cancer, oropharyngeal squamous cell carcinoma, breast cancer, gastrointestinal cancer, lung cancer, adrenal cortical carcinoma, prostate cancer, and head and neck cancer. Later states mebendazole might be an excellent candidate for the treatment of brain tumors because of its efficacy both when used as a monotherapy and in combination as an enhancement to standard chemotherapeutics and radiotherapy due to its effectiveness on tumor angiogenesis inhibition, cell cycle arrest, apoptosis induction, and targeting of critical pathways involved in cancer such as hedgehog signaling. And it had a graphic associated with this paper where it's kind of looking at what they found in each of these type of cancers, whether it be head and neck cancer, melanoma, gastric cancer, prostate cancer, brain cancer. Seems like it's been heavily studied for GBM and brain cancer, lung cancer, breast cancer, liver cancer, leukemias, ovarian cancer, adrenal cancer, thyroid cancer, and pancreatic cancers. So I'm going to spend probably the majority of the time in this video on this particular paper because it is very recently published and had some great graphics. So I wanted to give us kind of the current, most recent review of mebendazole's possible cancer fighting activities. And it was published in July of 2025, or roughly a year ago, and is titled Critical Dysregulation Signaling Pathways in Drug Resistance, highlighting the repositioning of mebendazole for cancer therapy. And it says in the abstract, cancer drug resistance significantly reduces the effectiveness of current anti-cancer treatments. Multiple dysregulated signaling pathways drive cancer initiation, progression, and related drug resistance. This highlights the need for developing new multi-targeting drugs that are more cost-effective, have fewer side effects, and remain effective against cancer. Drug repurposing offers a promising solution to expensive targeted therapies and helps overcome drug resistance. Mebendazole, albendazole, flubendazole, and oxfendazole are broad-spectrum antihelminthic drugs from the benzimidazole family. Therefore, mebendazole demonstrated potential in suppressing the growth of various cancer cells, both in vitro and in vivo. Consequently, we thoroughly reviewed mebendazole as a therapeutic option against cancer and related drug resistance. In this study, we identified mebendazole as a promising cancer treatment and works through multiple mechanisms such as regulating tumor angiogenesis, autophagy, apoptosis, modulating key signaling pathways, boosting anti-tumor immune response, and inhibiting matrix metalloproteinases activity, all of which are major factors in cancer drug resistance. Additionally, the development of new mebendazole delivery systems aims to address its pharmacokinetic limitations. While the anti-cancer effects of mebendazole are encouraging, further research is needed before it can be used clinically. Extensive data from in vitro, in vivo, and clinical trials support mebendazole's anti-cancer potential and highlight the need for innovative delivery methods, including polymeric nanoparticles, nanostructured lipid formulations, micelles, nanosuspensions, and beyond. And it had a nice graphic that was kind of highlighting all of the kind of major signaling pathways that are related to specifically drug resistance in oncology. And a lot of these signaling pathways we have covered in the past, specifically PI3K, AKT, mTOR, as well as HIF1, which is strangely missing from this graphic. In the paper, it says mebendazole employs several mechanisms to target tumor cells in cancer treatment. It inhibits growth and proliferation of cancerous cells by preventing microtubule formation. Mebendazole also suppresses tumor angiogenesis and stimulates autophagy in cancer cells. Additionally, mebendazole triggers apoptosis in various cancer cell lines by balancing apoptotic mediators such as BCL2, BCL2-associated X protein, BAX, P53, and X-linked apoptosis inhibitor XIAP. It also inhibits metastases, reduces the formation and size of primary tumors and prevents liver and lung metastases. Mebendazole induces the production of reactive oxygen species and DNA damage in tumor cells. Moreover, mebendazole, when combined with radiotherapy, enhances the toxicity of natural killer cells and decreases cancer cell transformation. It modulates several signaling pathways critical to cancer progression, including STAT1 and 2, E26-like protein, serum response factor, SRF, AP1, and c-MYC proto-oncogene, MYC, and MYC-associated factor X, MAX. Additionally, mebendazole boosts the efficacy of anti-cancer drugs such as cisplatin and docetaxel. Additionally, recent reviews point to the anti-cancer potential of benzimidazoles. However, in this review, we focus on the mechanistically based anti-cancer activity of mebendazole, since mebendazole addresses multiple mechanisms of anti-cancer drug resistance and appears to possess all the properties of an effective anti-cancer drug. We conducted a comprehensive review of its potential as a candidate therapeutic agent for cancer treatment. In a novel, in-depth review of the mechanistic repositioning of mebendazole and cancer drug resistance, highlighting innovative delivery systems. And basically what this paper does is it actually goes through each of the proposed mechanisms and explains how mebendazole interacts and makes all of these things likely possible. So it'll have a section on microtubule disrupting activity, inhibition of tumor angiogenesis, autophagy, synergy with radiotherapy, apoptosis, anti-tumor immune response, and decreasing matrix metalloproteinases 2 activity. And it then had a great graphic at looking at the activity of where mebendazole is directly inhibiting or downregulating certain processes that make cancer cells more likely to survive and withstand conventional standard of care treatments. Clinical trials of mebendazole in cancer. To date, various clinical trials have been conducted to investigate the efficacy of mebendazole as a potential treatment for cancer. In 2021, a single-center dose escalation study of mebendazole was conducted in 24 patients, 18 with glioblastoma and 6 with anaplastic gliomas, in combination with temozolomide. The outcomes showed a mean survival duration of 21 months, with 41.7% of patients remaining alive at the end of 2 years, followed by 25% who survived at 3 and 4 years. 17 patients taking more than 1 month of mebendazole from the date of diagnosis, median progression-free survival was 13.1 months, but 9.2 months for the 7 patients who received less than 1 month of mebendazole. Finally, mebendazole at doses up to 200 milligrams per kilogram showed long-term safety and low toxicity. During a phase 2A clinical study, mebendazole up to 4 grams per day and a serum concentration of 300 nanograms per ml was administered to patients with advanced gastrointestinal cancer. In individuals with advanced cancer, the personalized dosage of mebendazole was characterized by a favorable safety and tolerance profile. Mebendazole 200 milligrams per day was employed in individuals diagnosed with high-grade glioma who recently had begun undergoing temozolomide therapy. Other clinical studies have demonstrated the safety of mebendazole at up to 500 milligrams per day. Other clinical trials are planned for recurrent or progressive pediatric brain tumors, a dose of 500 milligrams per day. Cirrhotic patients with advanced hepatocellular carcinoma at the dose of 100 milligrams twice a day, and clinical safety and efficacy of mebendazole in gastrointestinal cancer. Conclusion. In conclusion, in vitro and in vivo, the clinical trial data provided ample evidence of an anti-cancer effect of mebendazole treatment and its related role in preventing cancer drug resistance mediators. This low-cost drug, or ideally low-cost drug, or should be a low-cost drug, characterized by completely stable pharmacokinetics and low toxicity, is a suitable and powerful option for drug repositioning in cancer treatment, both alone and in combination with existing standard therapies. However, other critical approaches are also employed in oncology drug repositioning, including molecular docking, genetic association, pathway mapping, signature mapping, retrospective clinical analysis, novel data sources, phenotypic screening, and binding assays. In this study, we investigated mebendazole in terms of its cancer treatment mechanisms and its role in treating various cancers through the regulation of tumor angiogenesis, autophagy, and apoptosis, as well as modulation of effective signaling pathways, induction of anti-tumor immune responses, and suppression of MMP2 activity. Such mechanisms are followed by the main anti-cancer potential of mebendazole on microtubule targeting. Despite the similar mechanisms of mebendazole and colchicine in targeting microtubules, they exhibit distinct selectivity, cellular uptake, and pharmacokinetics. Accordingly, possessing a higher volume of distribution makes a multi-organ toxicity profile for colchicine. Although we have now demonstrated diverse and interesting findings on the effect of mebendazole on various cancers, further research is needed in the future for its clinical applications. The findings of the present review indicate that the practical application of mebendazole in anti-cancer treatments will expand. Further understandings of anti-cancer mechanisms of mebendazole will pave the way for identifying the novel clinical relevance of mebendazole as an anti-cancer agent. Prospective research avenues should encompass comprehensive in vitro and in vivo investigations aimed at eliciting the exact signaling pathways of mebendazole, followed by tightly regulated clinical trials. The proposed investigation seeks to explore the potential anti-cancer properties of mebendazole in the areas of prophylaxis, mitigation, and therapeutic intervention for the diverse range of malignancies. Despite the promising findings on mebendazole oncology repositioning, some challenges and limitations remained, including poor solubility and slow disintegrating rate, which resulted in lower bioavailability. Furthermore, the variety of resistance mechanisms and variability in response to different tumor types due to differences in genetic profiles, tumor microenvironments, and drug metabolism between patients raises concerns about mebendazole's anti-cancer potential. These pharmacokinetic limitations urge the need for the development of novel formulations of mebendazole, such as polymeric nanoparticles, nanosuspensions, solid lipid nanoparticles, and micelles. To address the limitations mentioned above, combination therapies and patient-specific approaches would be critical for extrapolating mebendazole's anti-cancer effects into clinical success. I think after reading these papers, it makes a ton of sense why researchers, clinicians, and even patients are so eager to see mebendazole added to clinical practice in the coming, hopefully, months and years. But as with most of these things, the trials and studies take time to be done and for the data to be collected. And that can be extremely frustrating for patients who are in need of better therapies that make sense and that dovetail into the mitochondrial metabolic theory of cancer sooner than later. In the next video, we're going to talk about specifically the metabolic targets of mebendazole for the metabolic management of cancer. So if you like videos like this, please like, share, and subscribe. And until next time.